Okay, so this video is about blood types and how you can do Punnett squares to determine blood types of possible offspring. All right, so the first thing you have to know about blood types is you have to know the different phenotypes, okay, and that's the literal blood types that humans can have, and then the genotypes that correspond, okay? So if I tell you that somebody is type or blood type A, okay, then they can have two different genotypes. And if I tell you they're blood type B, there's two possible genotypes. If I tell you they're AB, that's another one. And then if I tell you O, okay, so these are your four phenotypes of blood type of humans. Now, positives and negatives have to do with the rhesus factor, and I'll show you that in just a minute. But right now, we just have to figure out the blood types and how you determine what type of baby that two parents can have. All right, so blood type has to do with the type of glycoproteins and glycolipids that a cell has on it, a red blood cell has on the surface, so that the immune system recognizes them as safe. So if you put type B blood into a person that has type A, their body and their immune system freaks out because the membrane proteins don't match what they should. So they look like a foreign substance, and so the body attacks them. So your blood type is very important to life. So with type O, we call these universal donors. And the reason we say that is because they don't have any markers to make them look like bad guys. So you can give blood to type O's. So type O's can give to type O's. They can give to ABs, they can give to Bs, and they can give to A's. So if you're a type O, they love to get your blood at the Red Cross because no matter what emergency occurs, then your blood is pretty safe to give to anyone. All right. Type AB, we call them universal recipients. So, universal recipient means you can receive. And so they can receive blood from type O. They can obviously receive blood from type AB persons. But since they have B blood cells, you can give them B blood. And because they have A blood cells, you can give them A blood. So they literally can be given blood from any person with their blood type. All right, so let's talk about when you're doing the Punnett squares, though, and I'll get a little bit more into the details of the membrane proteins later. But with the Punnett squares, there's two ways that scientists delineate blood type. Blood type is actually kind of cool because it's like codominance mixed with a little recessive. Okay, so in this situation, O is always recessive. Okay, he always loses to everybody. But A and B are codominant. So A cannot beat B if you inherit an A, and B cannot beat A. So they both show up. So a person who is AB blood, that means they received an A and a B gene. So one parent gave them an A allele and one parent gave them a B allele. And so we don't call that C, we call that AB. That means they actually have both. Now, some scientists and some geneticists write it like this. So they write a capital I, showing that that is a dominant trait, and then they use little exponents. Okay, so they use capital IB as one allele, capital IA is one allele, and then for O, they write a little I because that shows he loses to everybody. So I'm going to show you both ways that books write these genotypes. So AB has a B gene and an A gene. Type O, you can write it as two O's as long as you know they lose. But if you're using this system of I's, then you write it as little I, because they don't have any A genes or any B genes. All right, a person who's type B, they actually have two options. They can have two B's, or they could have inherited a B, and I'll write that as an O. Okay, so they could have inherited a B gene and an O gene, but B wins against O because O is recessive. If you're doing the I system, they would have a capital IB, or you could write it as a capital IB and then a little I. And then same thing with A. In the abbreviated system, they have two A alleles, where they got an A and an O because O is recessive. In the I system, it would look like this or like that, okay? So both systems work. I'm going to use this one, and then I'll show you an example when they use the capital I's. But it's the same deal. A wins if it's with O. B wins if it's with O. A and B are co-dominant if they show up together, and the only way you get type O blood is if you get two O alleles. So when you're doing Punnett square with blood typing, if I told you a type AB parent is crossed with a type O parent, they're pretty easy because the AB person has one A allele and one B allele. Type O blood can only have O alleles. So when you do the Punnett square for these two parents, this parent could have given the A allele or the B allele to their offspring because they only give half. This parent can only give the O, so I'll just draw two O's there. So when the two, we'll say this is mom, and then we'll make this dad. So when the egg has the A and the sperm has the O, that baby is type A blood. When the egg has the B and the sperm has the O, that baby's going to be type B blood. And then same here and same here. So this is a situation where an AB parent with an O parent has babies that their blood types don't match. So it's a 2 out of 4 chance of type A blood and a 2 out of 4 chance of type B blood, okay, or 50%. Now, when you are told the genotype of the parents, it's a little bit easier. So like if dad is BO and mom is AO, those are their genotypes. So that means the phenotype is dad would be type B and mom would be type A. And if you did the Punnett square for them, basically the same deal. One parent's alleles across the top, the other parent's alleles across the side, and then you fill in the boxes, A and B, A and O, B and O, and O and O. And in this combination of these two parents, it's a 1/4 chance of all of the 4 blood types. Now, if you don't know the parent's blood type, so if dad was type B and mom was type AB, she's definite. There's only one option for her. But dad could be two things. He could be BB or he could be BO because based on this table, and if you don't have this already written down, you should probably pause the video and write this down. But based on this table, there's 2 ways to be type B blood. So since I don't know, then I'm going to have to do 2 Punnett squares. So the first way will be if he is homozygous for type B. So that'd be AB, AB, BB, and then BB. Okay, so in this situation, it's 2 out of 4 chance that it's an AB blood and 2 out of 4 chance that it's a B blood. Now, if the parent is BO and AB, then we have this, okay, and then that's an AB, that's an AO, that's a BB, and that's a BO. So now the chances add in one new option. So we have a 1 in 4 chance of AB, 1 in 4 chance of A, but a 2 in 4 chance of B, type B. Oops, not BO, type B. All right, so when you are doing these, if I want all the options, then you would have to put them together. But in these situations, yeah, the 2 Punnett squares would work. So so in this situation, there's 8 boxes. So 2 and 3, so that would be a 3 out of 8 chance that their child has AB. This is assuming we don't know dad. 2 out of 4B and a 2 out of 4B. So that's going to be 8. So we've got 4 out of 8 that it's a B. And then we have 1 out of 8 that it's an A. Because if you look at it, there's one A, there's 1, 2, 3 B's, and then ABs, excuse me, and then there's 1, 2, 3, 4 type B. But that's a rarity. You just need to know how to do the Punnett square. All right, so now I'm going to show it to you with the letter I. Just to refresh your memory, this is the table. These are the genotypes if we're using the I system. And so I'm going to do a type AB with a type O parent. So if I'm using the I's, I do big I A, big I B, because those are the dominants. And then O is 2 little i's because those are the recessives. So I do the box the normal way. This parent has a dominant A, a dominant B, a recessive, and a recessive. So here we have big I, A, little I, so that would be blood type A because A beats I. Dominant B, recessive O, type B. Dominant A, recessive O, type A. Dominant B, recessive O, type B. So in this situation, A and A, so 2 out of 4 type A, and then B and B, 2 out of 4 type B. Okay, and that's how you do blood type. So you literally, it's Punnett squares just like we've been doing, but you just have to use a different set of alleles because this one has multiple alleles. All right, so it's not just a big A and a little a or whatever. It could be an A, a B, or an O, but you only inherit 2. So just because there's 3 alleles doesn't mean you get 3. It's just there's 3 options. Everybody gets 2. Okay, now, last thing I'm going to show you is plus and minus. So that's the rhesus factor. Plus is dominant and minus is recessive. Okay, and when I do the 4x4s, I'll show you how to combine blood type and do a prediction of the letter and the plus and minus. So a person who gets 2 pluses is going to be positive. If you receive a positive and a negative from the parents, you're still going to have a negative type blood and 2 negative. Let me start over. You receive a positive from each parent, you are going to have positive blood. If you receive a positive and a negative, you're still going to have positive blood because positive is dominant. If you receive 2 negatives, then you have negative blood. So some of you are A positive, some of you are AB positive, O positive, O negative, all those things. So this is how positives and negatives work. So same deal. If I have a mom she's positive positive and a dad that's positive negative, then she gives positives to the baby and he gives a positive and a negative to the baby. So the baby here, positive blood type, positive blood type, positive blood type, positive blood type. So even though he carries that negative allele, you have to get it from both parents. So having negative rhesus factor blood is pretty rare in most cultures. But in our dihybrid video, I will show you how to determine blood type and positive negative probability all at one time.